Okay, everybody. Good afternoon. Welcome back. Um, I guess you guys have been back for a few days. We've only just gotten back from Cognitive Neuroscience Society. Thank you for submitting all your second homework. It looks really good so far. All but one person has submitted it, so I'll just wait for everybody to submit it before I actually grade them. Um, so some of you have asked about the vision lecture. So as you know, your test is next week. All of the lectures so far should be online. Now, of course, there was Professor Kurtz came and gave the lecture on vision the day of my dad's funeral, and that was not recorded. And so some people asked for it to be recorded. So would you like fries with that? I have now uploaded my lecture on vision onto YouTube, and onto Moodle, so it's right there, along with all your other lectures that are on the second test. So it should cover roughly the same things that Professor Kurtz covered. There might be like little emphasis differences, but if you're in doubt, you should go with what's on my set of lectures, which is on YouTube. Okay, but today the goal is to finish talking about attention, and we were actually at the tail end of the previous topic, which was sensory integration, and so we talked about synesthesia, and that was right before the end of the last lecture before spring break. And I just want to talk a little bit about hemispheric asymmetry, which is really at the intersection of the work on sensory integration and the work on attention. And you'll see why in a bit. So when we're talking about hemispheric asymmetry, we're talking about differences between left brain and the right brain, right? So there's all this colloquial knowledge that, oh, if you're a left brain, you're more analytical, right brain more intuitive. Well, there's not a whole lot of scientific evidence for that. But that said, there are some differences in processing between people who do and do not have access to both halves of their brain at the same time. So what connects the two halves of your brain? Of course, it's the corpus callosum, right, which is this white matter bundle that connects your left hemisphere to your right hemisphere. Now, just looking at the anatomy of the corpus callosum, right there in the front part of this is the genu, and then most of it, the longitudinal part is the body, and then the posterior part is the splenium, right? So this is just the three components of the corpus callosum. And what's curious about the whole part, the whole three components of the corpus callosum is you could have people who either are born without a corpus callosum or have had a corpus callosotomy, which is actually a resection or removal of the corpus callosum. And this can be done either for the whole corpus callosum or just for part of the corpus callosum. So this here is the corpus callosotomy sparing the splenium. So it's quite too bright here, but you can see, you can see on the slides that generally this person, gosh, can you see it at all? If you don't mind closing that one, I think that might help a bit. This room is just not great for slide presentation, is it? But you should be able to see that this person doesn't have the corpus callosum except for the splenium part, right? And so that would mean that most of the left and right hemispheres, thank you, that does help a bit. Most of the left and right hemispheres are able to communicate directly with each other, but the posterior part is there's still intact connectivity. So there are people who are walking around with agenesis of the corpus callosum, right? So that means just people who are born without a corpus callosum. And they're normal just like you and me. I've actually seen one of these cases myself. You wouldn't know it until you look at they go into the MRI as a normal human subject who are participating in a study, and then you find out afterwards that there's just never been a corpus callosum that's there. But while most people have an intact corpus callosum, there are some people who have had it removed, and this is usually due to epilepsy, right? So when you have epilepsy, part of your brain starts to seize up, right? And if medication doesn't really control it, a neurosurgeon might suggest a callosotomy, which is a section, right? So cutting off part of the corpus callosum so that seizures that are starting in one hemisphere cannot just spread to the right hemisphere. And so the corpus callosotomy, or the lack of a corpus callosum, is one of many types of syndromes that are together known as disconnection syndromes. Right? So that just means a hypoconnectivity, so a lack of connectivity. And usually you get a bag of neurological symptoms that are caused by damage to your white matter pathway. So these are known as split brain patients, right? So who either have agenesis or callosotomy in the corpus callosum. And so we've already talked a little bit about alien hand syndrome. So remember when we talked about motor control, we showed a video of a woman whose hand was doing things that she didn't really voluntarily do. And so that can result from lesions in your motor control areas, right? So including the supplementary motor area especially. But that, it turns out, can also result from lesions in the corpus callosum. Then people with disconnection syndromes also show difficulty in language tasks, and we'll see how that is in a moment. And then there are also actually a better performance in people with corpus callosotomy when encountering a dual task situation. Right? So if I'm asking you to count numbers while drawing a circle at the same time, while most people will show some kind of interference between the first and the second task, people who don't have a corpus callosum are actually a little bit better at dual tasking. So it's not necessarily always a bad thing, right? So people with corpus callosum agenesis are born without a corpus callosum, and so usually this just shows up in an unexpected finding. Right? So maybe somebody might complain about a headache or just be participating in a study like my subject was, and then you find out in an unexpected finding on a CT or MRI that this person just doesn't have a corpus callosum. And so not surprisingly, functionally, these subjects are less lateralized, right? So while most people use their left hemispheres for language, right? Most but not all people, we'll talk about that when we talk about language, it turns out that people with corpus callosotomy or agenesis of the corpus callosum tend to show more language activity in both hemispheres, right? So both left and right hemispheres tend to be active in response to language. And again, like I said, dual task situations, right, when you're given two cognitive tests at the same time, while most people show a detriment in performance, which is an effective inhibition, people without a corpus callosum show a lack of inhibition, right? So they're actually able to do both things at the same time. However, when you show unilateral presentation, such as visual or auditory presentation, to people who have corpus callosotomy, that actually shows interesting effects. So that means that when you're presenting something only to the left visual field, right, which then goes to the right visual cortex, or only to the right visual field, which then goes to the left visual cortex, that results in some very interesting findings. Yeah. I'm sorry? Oh, it's really very rare. Yeah, it's less than 1%. Yeah. So how are these tested, right? Well, there's old-fashioned ways to do this, and there's newer ways to do this. Newer ways to do this, of course, involves some kind of computer screen. Older-fashioned ways to do this, tachistoscope. I'm not even sure. Nobody uses this anymore, but it's just something that can briefly flash a visual stimulus to one side of your visual field at once. And Mike Gazzaniga, who was a keynote speaker at the Cognitive Neuroscience Society and also one of the authors of one of the very commonly used textbook of cognitive neuroscience, supposedly he coined the term cognitive neuroscience, but he's really one of the most famous cognitive neuroscientists alive right now, and he did a lot of this kind of split brain work. So rapid visual presentation of stimuli in a lateralized manner towards people who don't have a corpus callosum. So that's what Michael Gazzaniga looks like. Right? He looks a bit older now, right? And so this is a nice video that will show you what one of these subjects did.
Information from each side of the body goes to the opposite side of the brain. And many functions are performed half on one side.
Just pause that and make sure that my sound is coming out of there.
But certain tasks are committed much more to one hemisphere, especially in human beings. The right hemisphere is specialized for spatial judgments, while the left can describe its perceptions in words. Normally, these two specialized halves work together as one.
I know the left hemisphere and the right hemisphere now are working independent of each other, but you don't notice it. Now, you just kind of adapt. It doesn't, you don't have any feeling different than it did before.
The corpus callosum, a huge bundle of files, connects the two halves of the brain. When it's damaged, in Joe's case through surgery to remove epilepsy, then it almost seems that there are two separate people sharing a single head.
What we can do is play tricks by putting information into his disconnected mute, non-talking right hemisphere and watch it produce behavior. And out of that, we can really see that there is, in fact, a reason to believe that there's all kinds of complex processes going on outside of his conscious awareness. Let that free. Now I'm going to show you some things. I just want you to see. Ready? Right. When Joe focuses on a point, everything to the right of the point goes to his left brain, the dominant hemisphere for language and speech. So we can see here that when we flash a word or a picture, Joe is easily able to name it. Close your eyes and let you do a little work here. Okay, what do you got there? Okay, very good. Now, when a word or a picture falls to the left of a fixation point, that information goes to the disconnected right half brain. And as we can see here, Joe is unable to name it. Joe is able to draw the picture with his left hand, the left hand getting its major control from the right half brain. Good job. What do you think? So even though he can't name it, his left hand is able to draw out the picture stimulus of the picture or word that we presented to his right half brain. What did you see? Close your eyes and draw with your left hand. Let it go. What Joe, patients like him, or many teaches us is that the mind is made of constellations of independent, semi-independent agents, and that these agents, these processes can carry on a vast number of activities outside of our conscious awareness. Even though that goes on, there's some final stage, or some final system, which I happen to think is in the left hemisphere, that pulls all of this information together into a theory. It has to generate a theory to explain all of these independent elements. So, and that theory becomes our particular theory of ourself and of the world.
From impulses leaving the eyes to a theory of ourself and the world, we're beginning to learn how the cerebral cortex gives us our understanding. Francis Crick, famous for his contributions to molecular biology, is now fascinated with perception and with the relationship between the brain.
It's pretty amazing stuff, huh? So what exactly was going on here, right? So let me just use this diagram and gesture a little bit, right? So when words were presented to the right visual field, which means to the left brain is good at language, so left brain is able to read the word and is able to name the object. When words were presented or objects were presented to the left visual field, which then goes to the right brain is not as good at language, and so the person is not able to name what they saw, However, because the right brain also controls the left hand, the subject can use their left hand to draw the object that they saw. Right? So you're presenting different information to the left and right brains, and so it's almost as if your left and right brains are acting as different people if you don't have a corpus callosum that's connecting that information. So you should also note that the optic chiasm nevertheless has to be intact, right? And that's how the visual input gets from the left eye to the right brain and vice versa. Cool, right? So I think there's something really profound about what Gazzaniga says at the end, right? There's a lot of semi-independent processes that might be going along in your brain unconsciously, and so much of processing is unconscious. Your conscious awareness is really only the tip of an iceberg in a way. And when you have these deficits of sensory subsystems, such as corpus callosotomy, it's quite informative of what kinds of processing might be happening at the same time. And then, as we'll see more today, you do get these individual differences in perception that are actually quite common. So there's not necessarily one right or wrong way of looking at or seeing stimuli. So let me move on to more about attention. And so the classic work on attention comes from William James. Some people think of him as the founder of psychology. Right? And he's written a lot about a lot of things, especially about attention. Right? So this is a quote from William James's Principles of Psychology, in which he says, "Everyone knows what attention is," which turns out to not be true. It's the taking possession by the mind of one out of what seem like several simultaneously possible objects or trains of thought. And it implies withdrawal from some things in order to deal more effectively with others, and is a condition in which has a real opposite in the confused or dazed or scatterbrain state, which in French is called distraction. And does anyone speak German? In German is I'm not going to try. So not attention, I guess. And so I think that's quite a nice definition, even though, and it does cover a lot of how people think about attention these days, but it doesn't really explain everything. So let's talk about the different theories of attention and how we've thought about stimulus processing and selection. And then I want to go through a bunch of different paradigms and findings from EEG studies, there's one single unit recording study in there, but mostly also from functional MRI studies. And then I want to talk. I definitely want to get to the end of this, which is about disorders of central attention, and I think they are actually a little bit comparable to the corpus callosum deficits that I've just shown you. Okay, so let's quickly go through these. So, how have people thought about attention? We know that it has something to do with paying more resources, or paying more mental or cognitive resources to some stimuli as opposed to others. Maybe it has to do with selectively attending to one thing over others, or maybe it has to do with different stages of processing in the brain, right? So there's a set of theories that are talking about where in the perceptual to cognitive pathway attention actually starts to operate. Right? So if you're one who believes that attention starts to operate early in the perceptual cognitive pathway, that's an early selection theory. Otherwise, it's a late selection theory. Right? So that's early versus late selection, and we'll talk a little bit more about that in a bit. Then there's also the idea of different kinds of sources of attention. Things that, is attention something that grabs you, or is attention something that you decide to pay? Right? So if you're one who believes that attention is something that you decide to pay attention to something, it's a voluntary thing, that's an endogenous theory of attention. In contrast, if you're the type that believes that attention is something that grabs you, such as when somebody is calling your name in the field, you have to kind of orient towards it. That is another kind of attention, but that's exogenous attention. So that's endogenous versus exogenous attention. Then there's the spotlight model, which mainly comes from the visual attention literature, but that has to do with how you can focus your attention within a space. right? So one model of that is, to think about attention as if it were a spotlight, right? So I'm looking at a crowded scene of lots of different students right now. I could decide to focus my attention on one of you, even though I don't necessarily have to look at you, right? So there's a spotlight of attention, which can operate separately from vision itself. Then there's the idea of feature integration. And this was really influential from Professor Anne Friesman, who actually passed away earlier this year. And that's the idea that in vision, you use attention to combine different features in space, or in the field that you're among the stimuli that you're looking at. For instance, if you're just looking at this slide right now, this number 4, right? It has a certain shape, it has a certain color, right? And then it has certain lines or certain junctions. Those are all different features, right? And so you need to use attention to combine the features of the lines and the shape and the color. Right? So if you had no attention, you wouldn't be able to integrate those features. And then finally, and this is getting really popular right now, there's the idea of temporal attention. Right? So in contrast to attention operating over space, or over certain visual components or stimuli within space, some people think that attention operates over time. Right? So while you're walking around or just being yourself, your attention could be sampling your visual or your auditory space in a rhythmic manner. Right? So in that sense, at some points in time, you might be paying better attention to sound than at other points in time. So there's a temporal fluctuation in how one pays attention. So that's kind of an overarching view of all the different theories of attention that's out there. So let's talk a little bit about these early versus late selection models. And this is straight out of your textbook, right? So what do I mean when I talk about the perceptual cognitive pathway? Perception means taking sensory input and then making sense out of it. Right? So sensory input means your retinal image, your cochlea, vibrations on your eardrum, right? And so those are sensory inputs, which go through several levels of sensory and perceptual analysis, and then higher level analysis until you have some kind of central executive function or some kind of CEO in your mind that is deciding what to do or how to respond to your stimuli, right? And this is a view that a kind of theme that comes through your second unit, right, from vision onwards. You use top-down control at the level of the executive functions, but you also have bottom-up information that comes from your sensory input. Right? So this interplay between top-down and bottom-up information is really one of the themes of this course, and also especially of sensory processing. So the earlier selection theories of attention pertain more to the sensory side of things, whereas later theories of attention pertain more to the top-down or the executive side of things. So that's a vertical way of looking at it. This is a horizontal way of looking at it. It's pretty much the same idea, right? So you get a bunch of sensory inputs, and you have to have some selective filter, right? And which then selects what among your sensory inputs you need to pay attention to. And that makes sense, right? If you've got a very crowded visual scene that you're looking at, right? You really want to actually just focus on one person, you use your selective filter to just select a certain channel, and then you can devote more attention to that limited channel, and then that process channel then gets more fully processed, and then higher processing operates on that to generate a response. So that's an early selection model. You can also have a relative attenuation, or kind of a softer version of the early selection model, right? And so instead of having a purely selective filter that just only selects one channel and attenuates everything else, you can have a relative attenuation model, right, where some things are better attended, right, or kind of emphasized in your mind, whereas other things are just a little bit less emphasized in your mind, right? So the difference is that you're in this model is that you're not fully filtering out all the other channels, but you do still pay more attention to the selected objects in order to generate a response. So let's move on to a bunch of different paradigms and findings. Right? So some of this comes from very, very classic work in the 1950s, part of what's called the cocktail party problem. Right? So what's the cocktail party problem? So what's a cocktail party, right? Or a pizza party, or a wine and cheese party? It doesn't necessarily have to involve cocktails, but it's just about when you have a crowded environment, such as a cocktail party, and your brain is able to hone in on one source of sound in your environment. Right? So when you're at a party, there's lots of, maybe some waiters going around, maybe some drinks being passed out, maybe some lots of people talking all at the same time about all sorts of different things. What the cocktail party problem is the idea that human brains are able to selectively hone in on one aspect of this crowded stimulus scene, especially in audition. And this is actually a really hard problem. It turns out that computers can't do this, right? So if you take a bunch of people talking at the same time, and you tell a computer to just pick up this voice, there's no computer right now that can do it. But our brains can do it. So how do we do it? Right? So that's the process of selective attention. And we know that selective attention is how the human brain can solve the cocktail party problem. So studying selective attention might be able to help us, design better models to automatic speech transcription, for instance. And so there are a couple of different ways in which you could pay selective attention, right? So one is endogenous, right? So in a crowded cocktail party, you can decide, okay, I'm going to voluntarily listen to what this person is saying, right? And you don't have to move towards that person in order to still attend to what they're saying. And so that's endogenous or voluntary attention. But you could also have exogenous attention, right? So that's the idea of breakthrough of the unattended. So you and I could be having a nice conversation right now, and then somebody else over there, if they mention my name, suddenly I'm like, oh, what's happening over there? So that's a breakthrough of something that was originally unattended. And so that's exogenous attention, right? So that's when something that is not attended is suddenly takes over attention. So this cocktail party problem was studied using a bunch of pretty classic paradigms from the 50s. And so, one of these is called dichotic listening test. It's really quite simple. You're listening to two streams of speech at the same time. Right? And so these are two streams of speech that are being said to your left and right ears. And you're just supposed to shadow the speech. And shadowing just means you're supposed to repeat one of the sentences that is being spoken to your left or right ear. Right? So they'll say, for instance, in this trial, this woman is trying to shadow the left input. Right? So this dichotic listening task is a pretty kind of general, commonly used technique of studying cocktail party problem. Right? So successful shadowing means that you're able to attend, right? But sometimes you might end up with a person reporting a word from the other stream. And so that would be an example of failures in sustained attention. In vision, we also have a number of quite well-established paradigms, especially some that look at the spotlight model of attention. And so a pretty classic paradigm is when you're looking at, you're given an array of different objects, such as letters or numbers, right? And you're always supposed to maintain fixation, which means you're supposed to keep your eyes in the middle of the screen. Right? So keep staring at this red dot here. And then at the same time, you're going to be asked to pay attention to all these different objects on your screen. And afterwards, you might be asked, was there an A in your attended area? Right? So that's another classic task of visual attention. And then another classic task is the spatial cueing paradigm, right? So that's where, again, you have to maintain fixation in the middle of the screen, and then you get a cue, and that cue so this is the cue, the arrow here is cueing you towards attention in the right side of the screen, right? And so sometime after this cue, you're given a target. Right? So cue and then target. And your job is always to say to just press a button whenever you saw a target, or say where the target is, or maybe it's either black or white or something, and you're supposed to say what it is. And so in some cases you can have valid cue, right, which is in which case the cue tells you information about where the target is going to be. Or you could have a neutral cue, right, where the cue is neither telling you about the correct answer or the wrong answer. Or you could have an invalid cue. Right? So in this case, the cue is telling you that the target is going to show up over there. Instead, the target actually shows up on the right side. Right? So, so the difference in reaction time between the validly cued and the invalidly cued trials is the cost and benefit of attention. Right? So that's how you measure attention using the spatial cueing paradigm, just by comparing the valid and the invalid cues, and sometimes with the neutral cue in the middle. Now, this is a very well-established paradigm, and so people have not only looked at the behavioral reaction time and response to these paradigms, but people have also recorded many different event-related potentials in response to these different kinds of paradigms. Right? And so in general, we talk about event-related potentials as certain components of brain activity. And so we label these with the polarity, which is either positive or negative, right? And then with latency, which is just how long it took for the event-related potential to show up in the brain. And so a couple of examples of this. An auditory N1P2 just means that there's a negative waveform around 100 milliseconds because N for negative, right? And 1 just means 100. And then that's followed by a positive component at around 200 milliseconds after the onset of the stimulus. And another one that's quite a common one is called a mismatch negativity, which is a negative waveform at around 200 milliseconds after the onset of anything that's unexpected. And so that's an index of how sensitive you are to the perturbation in your environment. Okay? So the more sensitive you are, the larger mismatch negativity you tend to observe. Then another one, another of these event-related potentials is the P300, which is a positive waveform, because P is for positive, right? And then 300 milliseconds after the onset of the stimulus, and that is always observed in response to task-related processing. So so what does that mean? So a pretty standard kind of task might be, I give you a bunch of visual pictures of Wesleyan basketball players and Amherst basketball players, and then Wesleyan football players, and then Amherst football players. And you're supposed to press a button whenever you see anyone from Wesleyan and not Amherst. Right? So whenever you press that button, right? So whenever you're doing something that's task-related, 300 milliseconds after that button press, after that stimulus comes on, you get a positive waveform in the event-related potential. So that's why it's called a P300. It's a positive waveform around 300 or longer milliseconds after the onset of the stimulus that is task relevant, right, that you're supposed to respond to. And then another of these, the last of these event-related potentials I'll talk about is the N400. And so you get the drill by now, right? N means negative, 400 means 400 milliseconds. And that is sensitive to semantic incongruity. So the classic example is if I say something like, I take my coffee with cream and socks. That was a little unexpected, right? Usually you expect cream and sugar after I take my coffee with cream and. But of course, so the finding is that 400 milliseconds after the word socks came up, you got a negative waveform in the brain. So that's a kind of double take that your brain does that is sensitive to the semantics of language. Right? So it turns out that if I say, I take my coffee with cream and NutraSweet, NutraSweet is not as unexpected as socks to go in coffee, so you don't get as big of an N400. Right? And so these are very important common findings that, like if you go to a Cognitive Neuroscience Society, as we did, that you'll see a bunch of different posters that look at these kinds of event-related potentials and how they're modulated by things like attention. So we'll move on to how things are affected by attention. So what's quite standard finding is that attention changes, or especially usually enhances, the amplitude of these event-related potentials. Right? So if I give you a few things to attend to, I tell you to pay more attention to, let's say, Wesleyan football players than Wesleyan basketball players, but just respond to anything that's Wesleyan, you will get a larger P300 effect for the attended object than for the unattended objects. And in the auditory world, you also get some enhanced event-related potentials. So again, this is you doing a dichotic listening task now, but instead of words, you might use something more kind of basic, like different tone pips, and different tone pips. So beep, 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 they could be played to your left and right ears. And let's say if the task is just to detect whenever there's a softer tone, and maybe you could be asked to detect only the things on the left ear or the right ear. But what the findings generally are when you're recording event-related potentials while the brain is doing these simplified dichotic listening tasks is that the N100, right, which is this negative waveform 100 milliseconds after the sound comes on, that amplitude of that N100 is larger in the attended channel compared to the unattended channel. So channel here just means ear, right? If I tell you to attend to things that are coming to your left ear, right, you get an N100 for any sounds that are coming to your left ear. You get an N100 for any sounds that are coming to your right ear, but the N100 is bigger for the attended ear than for the unattended ear. Okay? And so a difference wave just means the attended minus unattended, and so you can see that there's this higher amplitude right around 100 milliseconds, and you can localize that to different parts of the head, and generally the results are centered over the auditory cortex, which is where the N100 is. But attention doesn't just operate over the N100 and the P300. It's also shown over the mismatch negativity, right, which is around 200 milliseconds. And it's also observed in the P2050, which is about 20 to 50 milliseconds after any attended sounds. So, so I think the upshot is that we see effects of attention. So a while back, when people were still debating whether early attention is a result of early selection or late selection, people thought that if you saw event-related potential effects in earlier time windows, that should be a marker of earlier attention, whereas if you only saw them in late time windows, such as P300 and later, that should be a support for later selection. But it turns out that you do see event-related potential differences for both auditory and visual paradigms from quite early on, like around 20 to 50 milliseconds after the onset of the stimulus, all the way until about 300 or 400 milliseconds. So I think the most general model now is that you have some kind of attentional control network within your brain, right, that is controlling or acting upon the bottom-up stimulus processing. Right? So this is a very general, this blob is just a very general representation of the brain where the dorsal part is generally representing attentional control network, which might be operating upon some bottom-up stimulus processing areas. But we also don't just have a single area of, or even a set of areas of the brain for attention. It turns out that attention actually modulates the processing of sensory cortices themselves. Right? So this nice paper looked at a functional MRI paper looking at how the auditory cortex would respond to sounds while the brain is trying to pay attention to sounds, as opposed to when it's trying to pay attention to sight, to visual information. Right? So these people were given auditory information and visual information at the same time, and so there are these audio-visual discrimination tasks. But what the researchers were particularly interested in, they don't actually care about the visual cortex at all. They only care about the auditory cortex, but they just wanted to see whether your auditory activity would differ depending on whether you're paying attention to the auditory modality or to the visual modality. And so here they are just looking at the auditory cortex, so in the superior temporal gyrus and the Heschel's gyrus. And what they're seeing here is the SDA stands for stimulus-dependent activity, right? So which is the activity that's elicited by the auditory stimulus without attention being directed at it. So when attention is directed away from the auditory stimulus at the visual stimulus. And compare that to additional attention-related modulation, or ARMs, which is just an enhancement that turns out to be lateral to the stimulus-dependent activity. So even within the auditory cortex, but slightly lateral to the areas that are primarily driven by just the stimulus activation themselves, you do see a modulation that is due to attention in the auditory cortex. And not only do you see this in these patches of brains that we could capture in awake behaving humans, it turns out that even at the level of the single cell, right, you can actually observe tunings for receptive fields that are modulated by attention. Right? So this kind of receptive field for stimulus orientation is something that we talked about in vision. Right? So hopefully Matt also talked about it. I definitely talked about it in my lecture that I recorded. But what you can see is that cells that are at the level of the visual cortex, or primary visual cortex, have orientation sensitivity. Right? So one cell might be optimally sensitive, right? So it fires the most when it's seeing an edge that's vertical, right? And it fires next most when it's seeing an edge that's close to vertical, and not much at all when it's seeing an edge that's not vertical. Right? So this is stimulus sensitivity, right? And so each cell has a receptive field for stimulus orientation. But what this very nice paper observed was that the same cells can actually fire more when a certain orientation is attended. Right? So you could design a task such that the monkey, right, is more sensitive to, or is paying attention more to certain orientations than others. And it turns out that you can get attention-related enhancement of these single unit cells in NV1. So that's a bunch of evidence showing that attention operates at various levels. Right? You can think about attention as this overall network, but it also operates on the sensory cortices themselves, and they also operate at individual cell levels. All right, so let's move on to talk about what happens when you have problems with attention. And when you're talking about problems with attention, I know a lot of you have heard about ADHD, right? That actually is more of a problem with executive function, which we'll talk about later in the semester. But what I'm talking about right now is actually some disorders of central attention, mainly due to a brain injury or tumor or some kind of brain trauma that results in a profound inability to pay attention to stimuli. And so we're not just talking about people who have trouble focusing, but we're talking about some neurological problems that are much more pervasive. And the first of those is neglect. So neglect, I'll show you this video that's which should be illustrative. And then we'll talk about some neuropsych testing studies that have actually been done to show that these problems are really about attention and not just about perception. And then we'll talk about the brain underpinnings of that and what it means for studies of the brain. Oops. Codec unavailable. Oh. Okay, not sure what's happening there. Do I have my other videos? Okay, this one seems playable. Okay. One second. Okay, so I think I'll just demonstrate it. So these are neglect patients. Neglect is a constellation of attentional disorders that arise from lesions mainly in the right hemisphere. Right? And so when you have these lesions in the right hemisphere, you end up not being able to pay attention or just ignoring half of your visual field. So the video that should have shown is when a person is neglecting half of their visual field, so the right hemisphere pays attention to the left visual space, and so they're not paying attention to the left visual space. So first of all, they show a posture sort of like this, and so they're neglecting this side. So they're only paying attention to the right side, and then Jim, can I get you to come up and like take a pencil, take a pen, and like wave it, and my left side. So that's what the person is supposed to have done. Thank you. Except they were using a $20 bill instead of a pen, which makes things a lot more motivating, I guess. Yeah, so that's what the person is supposed to have done. So how do we design some tests to tell that they're not just blind on one side of their visual field, right? So first of all, we have the line bisection test. Right, so I'm going to ask for one volunteer. Any volunteers? No? Okay, thank you. One up. So can you please bisect this line for me? Thank you. That's very carefully done. Yes, stay up here because I got more for you. So you can see how he just nicely partitioned. What's your name?
Matthew.
So Matthew so nicely partitioned that line into the left half and the right half. Now, in contrast, this is what a person with neglect does, right? So they're not just not seeing the left half of the line; they're neglecting. They're just forgetting or not thinking about the left half of the line, and they're only treating the object as if it's only got a right half. Now, okay. So now what I'm going to do is called a line cancellation task. I'm going to erase this. And I'm going to give you a bunch of different lines. Can you please cancel all these lines? So just draw a line through all these lines. Yep, you're doing the right thing. Okay, thank you. So you can see how so Matthew has very carefully put a line through each and every single one of these lines. Right? So the whole field of lines are canceled. Now, in contrast, this is what a patient does. Right? So so first of all, they're neglecting half of the lines, and so they're only attending to the right half of the lines. And then furthermore, so this is so some of the lines he's drawn twice, and that happened because he went over a bunch of just the lines on the right, and then he said he was done. And then the experimenter said, "Are you sure? Do you want to double check?" And then he went back and did the same ones again. So again, profound neglect of the left half of visual space. Oh, this one's a fun one. I'm going to draw you a picture, and I'm going to ask you to copy it. Okay? So I'll draw it on the top half of here, and you can draw the bottom. Okay? So hands, which goes into, you don't have to be super careful about it because we don't have slides, but this is just an example of the kind of thing that you might be asked to do in the site. Thank you. I think that's plenty of work for you. Good. Thank you. Okay, so that's a very nice copy of the picture. 3 people, each with left and right arms. This is what So on the left we have on the top we have the specimens, and the bottom we have the performance. And quite striking, right? So the patients only drew the left persons, and then only the right persons, and then only the right half of each person. So that means that there's object-centered as well as spatially centered attention, right? That is both of which are neglected in the left half of visual space. Right? So same goes for the seeing here, right? Instead of seeing a nice picture of a house with left and right fences, they're neglecting the left half. This last one is really quite interesting. So if I were to ask you to draw a clock, you can think about how you would draw a clock, right? I can just do one because that's fun. So I'll think about drawing a round thing, and I've got my 12, and I've got my 6, and I've got my 3, and I've got my 9, right? And let's say if I'm supposed to draw 6 o'clock, I'm going to draw a short arm here and a long arm here, right? And maybe I'll then fill it in so that I've got all my numbers, right? So, it's kind of fun, not so surprising, nothing surprising there. I don't have neglect either, but so this is a neglect patient doing the clock drawing task. Hopefully this will work.
Just put the numbers on it.
I'll have it done.
Yeah, where the numbers go.
Amazing, isn't it? So not only did she squish all the numbers to the right side, so neglecting half of the space, she also only wrote the 2 out of 12. So there's object-centered attention, and then there's more spatially scene-centered attention. So if you're asked to draw to indicate 6 o'clock on this clock face, this is what one neglect patient did. Doesn't look like 6 o'clock at all. So what kinds of neuroanatomy gives rise to this? Right? So in general, right-sided lesions are more common, and there's a distributed attention network, right? And so this is kind of why the textbook showed the attention top-down control network to generally involve the dorsal parts of the brain, because people with neglect generally have missing lesions in the dorsal part of the right hemisphere. So that includes the temporoparietal cortex, including frontal eye fields, which are in the frontal lobe, kind of close to the motor areas. But then sometimes you also see some subcortical damage, such as thalamus and basal ganglia, and the cingulate cortex. And this is mostly due to right hemisphere damage. And most common cause, stroke. Right? So if you have a stroke in your right middle cerebral artery, that will likely affect the right parietal lobe, including some temporal areas. And this is not so uncommon. Right? And it's important to establish that this is not due to sensory loss. Right? So it's not just that their visual cortices are not working. Yeah? Aphasia. Aphasia, which is the loss of language. Yeah, and we'll talk about that. So for the most part, like the vast majority of left parietal damage and frontal damage leads to, well, motor problems, which could be lateralized, but the left hemisphere damage is much more likely to give you language problems because language is in the left hemisphere, and these attention problems are more likely right hemisphere. Is there another question over there? Oh, just stretching? Okay. All right, so this is just a 3D reconstruction of a patient, the brain of a neglect patient, and not super clear in here, but the lesion is right parietal lobe. So this gives rise to a number of theories about how attention works, right? And I think the most kind of influential of these is about, and obviously there's something asymmetric about our brain, and it's something asymmetric about how our brain accomplishes attention. Right? And so the dominant theory is about a global versus local difference. And so it might be that if you have you obviously need your right hemisphere, especially parietal lobe of the right hemisphere, to pay attention to your full space. Right? But your left hemisphere can also pay attention to some part of the visual space. Right? And this you could see from those Gazzaniga studies, right, where they were showing lateralized input to the left and right hemispheres. So your left hemisphere is able to detect information from your right visual field. But so, because your right hemisphere is doing, or is paying attention globally to your whole visual field, whereas your left hemisphere is paying attention locally to your right or contralateral visual field, if you have damage in your right hemisphere, you will end up neglecting your left visual field. So evidence for this global-local distinction also comes from these kind of fun types of stimuli. They're generally known as global-local stimuli. Right? So you can tell that there are a bunch of smaller E's that are the local stimuli, right? And they together comprise the S, which is the global stimuli. Right? And so these are pitting the global processing of the forest, or the S, against local processing of the trees, which is the E's. And so you could do this with people with left and right hemisphere damage. And so this is a very general classic finding. Patients with right hemisphere damage end up ignoring the local, okay? Whereas patients with left hemisphere end up, the global, whereas patients with left hemisphere end up ignoring the local. So people with right hemisphere injury are only replicating the local parts of the stimulus, right? Whereas patients with left hemisphere injuries are only replicating the global aspects of the stimulus and not the local aspects of the stimulus. Yes. And then, so in addition to neglect, you can also have some cases in which patients are actually able to process stimuli in space, but only one object at a time. So when you give people two objects at the same time, simultaneously, they are no longer aware of the two objects. So that's known as simultanagnosia. So this agnosia just means your knowledge or awareness of things, and simultanagnosia just means lack of awareness of simultaneously presented items. And so this comes from a kind of a right hemisphere damage known as Balin syndrome. It's a kind of a different variant of neglect. Right? So think of neglect as like a constellation of these different central disorders of attention, including simultanagnosia. So this here hopefully will work. So there's a neuropsychologist, his name is Bob Raval. He's a very famous neuropsychologist studying attention, and he's doing some testing on this one patient. So there should be more. So it's a short clip there. But you can see how when there's one object, he's kind of able to identify it. But as long as you have two objects at the same time, you can't see both at the same time. So that's, yeah. Yeah, now that's a very interesting question. Movement usually helps a little bit, but not completely. Right. So if a classic example, and that's a kind of bedside testing, that's clear, but a classic example is, remember that line cancellation class, or that I given where there's a bunch of different lines and you're supposed to cancel all of them? If the whole field is moving a little bit, you tend to cancel out a few more. Right? So it's that the action or the movement actually helps you regain or recover some part of your visual space, but not completely. Yeah, that's a good question. Any other questions? Yeah. Yeah, that's pretty classic. Where they just ignore the left half. And it's sometimes, I should also point out that these are pretty classic textbook cases. In real life, like if you were working at the VA, so let's say a lot of patients come back from that line, they might have had a traumatic brain injury from a blast happening on front lines. It's usually a changing situation, right? So at first, you might see a lot more acute symptoms, and then over time, you might get more and more of this, right? So the line cancellation sound is pretty classic, right? So the case that we saw on the slide is really bad. They're just showing, they're just noticing maybe like a bit of the lines, but over time they might regain and you might see more and more of the lines. Yeah. Yeah, we have actually Wesleyan alum actually working with these patient populations in the VA in Boston right now. So they're seeing many more cases of these. Okay, so so the simultaneous nausea. This is just a little bit of data from the lesion studies. So this is, I know we talked about the voxel symptom lesion mapping, or VLSM, where you're taking neuropsych populations and who have lesions in various areas of the brain, and you're doing a lesion overlap, and then you're relating that lesion overlap to differences in function. Right? So the upshot of this is generally you see problems with the posterior parietal lobe associated with simultaneous nausea. Right? So the guy that we just saw, right? Most common damage is in the right parietal lobe. Right, and so a much related idea is extinction. Right? So that's the idea. It's another of the constellation of problems that are in neglect. So patients with extinction can just detect a single stimulus at any spatial location, but as soon as you get two objects being presented at the same time, they are impaired at perceiving the contralesional item. So usually the left side is ignored. And again, it's right hemisphere damage, mostly parietal lobe. And so one theory proposed for explaining this is that you have competition between different stimuli that are competing for your attention. And while normal subjects have less difficulty attending, once you have a lesion, that leads to more of a competition or bias of the competition between your ipsilateral and contralateral stimuli. So I think that's all the stuff I've got for attention. I do want to spend a little bit of time talking about doing a little bit of review, but just as the last slide on attention, you can think about these theories of attention as ranging from earlier to later selection, and then also ranging from endogenous versus exogenous attention. And we went over a bunch of different paradigms, but they're mostly auditory and visual ways of isolating attention as an effect. Right? And so then we looked at ERP effects and MRI effects, and then also the neuropsych populations. Right? So the other thing I should really say about the neuropsych populations is that there's a constellation of symptoms. So you might start out with Bayley syndrome, which then becomes extinction, which becomes simultaneous nausea, or you might get one and then the other, or you might get both and then suddenly neither. Right? So they're not. It's not all or nothing. So think of them as like a group of things that might happen over different times of a person's brain injury. Okay. Any questions?